speak.
Okay. So, good afternoon, everyone. So, I would like to thank the organizers for giving me this opportunity to present this work in here. So, I want to show work that we have done. It is about the transient reverse fingers. So, this work explains some kind of instabilities that develop in confined geometries. So, to go deeper into it, I'll just like to explain some basics first. So, one of them is Safman-Taylor instability. Safman-Taylor instability is, so let's consider you have two fluids of different viscosities and you want to displace one fluid with another, and then you can have two conditions. One is when you can displace a less viscous fluid with a more viscous one, and you can displace a more viscous fluid with a less viscous one. So, when in the first case, when you are using more viscous, which is here yellow, to displace the less viscous, then you will see that this circle is growing circle. So, that means that any perturbation at the interface, they will get stabilized. But on the other hand, if you displace a more viscous fluid by a less viscous one, then interesting thing happens. So, here you can see that all the perturbations they grow, and any perturbations they induce these large scale growth and these repeated tip splitting also. These two are different geometries, which one is the rectilinear and another is the radial, but the phenomena that they shows are same. So the statement is that when a less viscous fluid displaces a more viscous fluid in a porous medium, the interface between the fluid becomes unstable, and you will see the perturbation growth and the large scale structure formation. So, since I am talking about the porous medium, so the porous medium is usually opaque. You can't see the flow inside a porous medium with a camera. So, in laboratory scale, a Hele-Shaw cell, which is just two glass plates separated by a very thin gap, they are used to study the flow in a porous medium, and it looks like this, that these two are the glass plates, and the gap between them is very smaller than the length of the plates. So, here is the top view of this plate, where you can see that there is one inner fluid and there is this outer fluid, and the interface will be unstable if this is more viscous. So, why this happens is because you can see here that let's imagine this less viscous fluid is displacing more viscous fluid. Imagine a perturbation that gets developed, and at the tip of this perturbation, the pressure will be highest, and so is the velocity, because it follows the Darcy's law, and this is why in this situation the perturbation grows. Okay, so why to study these kind of instabilities? So there are two different ways to think about it. One is that these are useful in industrial processes. So the use is that they are not wanted in the industrial processes. One example is the oil recovery process, where what you have is the injector and you have a oil collector. So, and here this is the porous field, oil field, where there is oil, and then the water is injected through the injector, and the oil is getting displaced and collected by the producer. But if the water is less viscous than the oil, then these kind of branch fingers will form, and as they form, the displacement efficiency or the oil recovery will be very less, and one would want to reduce this formation of these fingers in such cases. And another example is the carbon sequestration, where if a power plant is producing carbon, it don't want to emit the carbon to the atmosphere. It wants to they want to keep the CO2 at the underground stored, because so that they don't participate in the global warming. So in this case also, for the efficient storage of those, these fingers are undesirable. Another example where the fingers are desirable is when you have a mixing at low Reynolds number. So at low Reynolds number, only the diffusion will be there, but if these instabilities are there, they will make new interfaces due to these instabilities, and the mixing can be done at micro scale, micro channels. So the patterns that are formed due to this branching, those patterns are seen everywhere in nature. Some of the examples are the T branches, and this is land topography. This image is taken from the Google Earth, where this near the Uttarakhand and Himachal, these dendrite-like patterns can be seen, and the river branches also. And in human lungs also, you have the these air transmitters which forms this branch pattern, as well as lightning also form this branch pattern. So these kind of patterns are seen everywhere. So to study in our system, we are using aqueous cornstarch suspension. So to make a cornstarch suspension, you need the corn flour, water, and you just mix them, and you will see that these polydispersed irregular shaped particle, they will form a non-Newtonian fluid. So this picture is taken by K. Mithindran at SCM department, and this shows the cornstarch particles. So when they are dense enough, when you mix much more amount of those particles in the fluid, then you will see that this behaves like a solid. When you put a sudden impact on the suspension, it behaves like a solid. This is another example which shows that so this is a simulation paper where if the wheel is moving on the surface of the cornstarch suspension, if the rotational speed of the wheel is high enough, it will not sink. But if the rotational speed is low enough, it will sink in the cornstarch suspension. That means if you perturb this cornstarch suspension at high speeds, it will behave more like a solid, and you will feel higher apparent viscosity. So this property, where you feel the higher apparent viscosity with applied stress or the shear strain, is called the shear thickening property. So if you took a cornstarch suspension and put it in a rheometer plate, Sevanthi talked about a rheometer plate where you have two plates and you put the sample and you shear it. So if you do rotational test, you can get the viscosity versus shear rate, and you can see that the cornstarch suspension at low shear rate, it acts like a shear thinning fluid, which means that you will feel that its apparent viscosity has been decreased. And then if you increase the shear rate further, you will see that it is now getting more and more difficult for the sample to flow. So the viscosity is increased. But this transition where this shear thickening happens, it highly depends on the concentration of the suspension. If you have the higher concentration of the suspension, then this transition is more drastic than the lower concentration ones. And actually these transitions are also divided into two different parts. Both are shear thickening, but one is continuous shear thickening and other one can be called a discontinuous shear thickening. So to understand this, why this happens, the generally accepted model is that if you have dense particles in the solvent, so most of it is just particle only. So these particles can come into contact with each other when you shear it. So here you can see if you shear the bottom and top plate, these structures will form, and with increasing shear rate or with increasing Peclet number, you will see that this number or the density of these chains are increasing. And as you see increase, then it can also jam itself. So this is like a railway bridge where you have many contacts together, and then it can hold an enormous amount of stresses. So what we will do, we fill this cornstarch suspension in this Hele-Shaw cell, which is just a 2 glass plate, and we will displace this cornstarch suspension with water. So this is a picture where this is outer fluid is the cornstarch suspension, inner is just the water, the DI water, which is dyed also, and we'll observe that what happens at the interface. But what we are doing, we are not only observing just one interface, which I showed you earlier. Here the interface are two. This is the outer interface between the cornstarch and air, and this is the inner interface which is forming when we are injecting water. So it can be seen here that this is a representative picture of the pattern that forms, and you can extract the inner interface as well as this outer interface properties. So let's see what happens when we displace this cornstarch suspension at sufficiently different flow rates. So for low flow rates, what we see is that initially t equal to 0, we started injecting water. Then as water is going inside, due to the soft-mantle instability, the interface is unstable, and we see multiple tip splitting inside this side, and in multiple series of these tip splitting and these integrate patterns formation. And if we focus at the outer interface, we see that there is nothing interesting going on there. But if we displace the same suspension with the same water, but we change the shear rate or the flow rate, injection flow rate, what we observe is that in addition to this pattern at the inner boundary, we see that this kind of fingers are also occurring at the intermediate times, and they are very transient in nature. So initially there is nothing, but when the water is injecting, you see that these fingers form, and later time they are gone. To characterize these interfaces, we calculated the parameter of both the inner and outer interfaces. So we see that the inner interface parameters increase monotonically. These are the P-in. So these increase monotonically, but outer interface parameter shows this non-monotonic behavior because of the enhanced roughness due to the formation of reverse fingers. And one more thing to observe here is that the inner interface growth and outer interface growth, from this picture it looks correlated, because at the time when the slope of the parameter changes, it coincides with the maximum of the outer pattern parameter. And also we see that the number of reverse fingers at these points when the reverse fingers are maximum, the number of reverse fingers with flow rate they increase, but the average separation between them, that is lambda, that doesn't show any change with increasing flow rate. So there are these properties of inner and outer interface. I'll just explain. So inner interface shows multiple tip splitting, and formation of these branches. The morphology, which means that this morphology it is insensitive to the flow rate. They look really same with increasing flow rate. Inner pattern also shows this coalescence event where two fingers can merge with each other because there is a less amount of surface tension, so it can merge with each other. And parameter increases monotonically, which means that parameter, because the roughness is always increasing, tip splitting is always happening. On the other hand, at the outer interface, which we see the transient withdrawal of the suspension and the formation of reverse fingers, and the reverse fingers, the formation or the spatial extent of those reverse fingers are also dependent on the flow rate. They also are transient in nature, they fade away at later times, and their temporal variation of the parameter is non-monotonic. So we observe these fingers, and now we want to see that what are the many control parameters. So we started by changing the particle concentration, which means now we are using more dense cornstarch particle. Now there are more number of particles in it, and in this case, and here there are less number of particles, and we see that the reverse fingers are only forming for the denser side of the particle, which is 40 and 42% reverse fingers are forming, and for this it is not forming. And also when we change the Hele-Shaw cell gap, which is the gap between two plates, and then we see that the reverse fingers goes away with the increasing gap. So and also up to this point, all the patterns are formed when we displace the cornstarch suspension with water. But the another possibility is could the interfacial intention would play any role in the formation of these things. So we see that even if we use the mineral oil, which is an immiscible fluid with respect to the cornstarch suspension, this cornstarch suspension also shows these reverse fingers at the intermediate times. So these reverse fingers are present when you have high shear rates, or when you have high concentration, when you have a low Hele-Shaw cell gap or the higher confinement, and the effect of miscibility, they will form irrespective whether they are displaced by air or water. And there are two characteristics of these reverse fingers that we have identified. One is this: these reverse fingers are not always axially symmetric. So if the reverse fingers are forming only in one way, the inner pattern growth is also connected with that formation, and the inner pattern growth is less in that. So here you can see that R is the inner pattern radius, and NRF is the number of reverse fingers in that segment. So here this is divided into 8 segments. So if the higher the inner pattern radius, lower the reverse fingers. So that means these two are correlated. Other than that, when we change the CS suspension radius, which means that if I am filling the suspension till 5 centimeter in the Hele-Shaw cell or till 10 centimeter, the time when the reverse fingers reach their maximum, it increases monotonically as shown here. And from this time, so that this suggests that there is some information that is transmitting from the inner interface to the outer interface, and that is the stress, and we can calculate the stress propagation velocity from this slope, the inverse of this slope, and that is 4.52 centimeter. These numbers and these scales match closely with the previous study of Maharajan, where they have also studied the stress propagation in cornstarch suspension. So I'll try to finish it quickly. So what we are doing is that we also do some rheology experiments where we want to see what are the regime where our experiments are falling into, and what we see is that shear rates, which these shear rates are calculated from the pattern. So you have the finger growing, you can calculate its velocity, then you can get the shear rates by this formula. And what we see is that the shear rates lie in the shear thickening regime of the Cornstarch suspension. And at those regimes, what we see is that the normal stress, which is the which comes when you have which comes positive when you have a frictional contact between particles. So these are positive and these are quite large. So this suggests that at those shear rates, the outer fluid, that is Cornstarch suspension, that is forming the frictional force chain networks. So the mechanism that we propose is following: that the the transient stresses are transmitted through the system spanning network of frictional contacts, and then dilation happens, and then at the soft boundary means that at the outer interface the deformation happens, and then the surface tension acts as a restoring force and pushes back. Reverse fingers will then form, and then they will eventually fade away because of the rearrangement of the Cornstarch particles. So this was the study. I like to just go to the acknowledgement side first. So Professor Ranjini, Palak and Sayantan have worked with me in this project. I'd like to also thank my lab members for their useful discussions. So this is
Yes, thank the speaker. Questions?
So no, can you can calculate this roughness or growth exponent, things like that? And have you tried? I mean, is it known that whether it falls close to this KPZ scaling and KPZ university class and so on?
KPZ scaling I haven't tried. So you are talking about the how the inner interface growth. So that I haven't tried. Yeah, but I am trying it as of now on a different sample. But for this I haven't tried it.
And also, I think you said, but I missed it. Could you say again the cause for the non-monotonic growth for this outer perimeter? Why is it non-monotonic?
Yeah. So as you can see that so the perimeter is just defines the roughness of the interface. So the roughness is maximum when you have reverse fingers. So this is with time. You see that at initial time it is smooth, relatively smooth, and at the intermediate time these fingers form, and later it is all smooth. So the roughness is initially increasing and then decreasing. That's why it is non-monotonic.
Very nice talk. So one thing is you mentioned that how much you fill the suspension, like the Cornstarch suspension, that will also affect when you see the reverse fingering. Yes. So what is the smallest limit you have gone to? I mean, is it possible that your like inner and outer fingers will be very close to each other?
Yeah, so we have went to 5 centimeters. So we have filled the Cornstarch suspension to a diameter of 10 centimeter minimum.
Okay.
So that is actually enough to see the reverse fingers. But I think if we decrease it further, then we will not.
You won't see the reverse fingers at all.
I don't know. I haven't tried it, but I think it will mix quickly, and then I will not.
Oh, you won't be able to make it. Okay.
Yeah.
Any other question? Okay, if not, let's thank the speaker. And let's thank all the speaker of this session. And